Okay, I'm going to cut right to the chase. Bacteria is key to our health. And there's good bacteria and there's bad bacteria. The good bacteria create enzymes that break down food, that attack invaders, that create all kinds of different products based on your diet and what kind of an environment you're in, what kind of air you're breathing, all of that. And they're very, very specific. And they're created by specific bacteria. And if those specific bacteria are not in good enough quantities and thriving and eating and living and populating inside you, you will not get the enzyme that they are supposed to secrete. All your organs secrete enzymes. They don't secrete them by accident. They secrete them because there's bacteria living in there that secrete them as ribosomes. You see this? CAT scans, advanced type of X-ray, takes many images from different angles to create a detailed cross-section of the body. It uses a much higher radiation dose than a single standard X-ray. They're shooting a bazillion different shots and much higher, and this is ionizing radiation which kills bacteria. Now don't forget, this is ionizing radiation. X-rays kill bacteria by damaging their cellular components through the process of sterilization. They think this is good. The effect is used in food irradiation to eliminate foodborne pathogens. Well, they're the same pathogens that are living in your body, only they're good for you in many, many, many cases. There's bad ones. I'm not saying they're all good, but the ones that are good, if you kill them off from these radiation exposures, and they are ionization. And here's what happens from radiation. You get radiation sickness. Now listen to this now. It's also known as acute radiation syndrome. Well, what happens? You get popped with a nuclear explosion somewhere on the outskirts of it, and you get hit with a high amount of energy. All right? This acute radiation syndrome, a condition occurs when a person is exposed to a high dose of ionizing radiation in a short period. That sounds to me like very similar to a CAT scan. Now this is, and it talks about medical treatments using radiation therapy, exposure to nuclear weapons, accidents, medical treatments using radiation therapy. Now, you get nausea, vomiting, diarrhea, fatigue, and fever. Why? Why would that come to you very shortly afterwards? The symptoms vary depending upon the dose of radiation and the time since the exposure. Sometimes within like an hour or two, you just start throwing up and you have diarrhea and you just can't move. You fatigue, and all of a sudden you get inflammation and fever starts to kick in. They really don't know what causes this, basically. Now, as far as my research goes, if it killed a whole bunch of good bacteria in you, you're going to have all kinds of issues. And they say high dose of radiation can be fatal, obviously. With prompt treatment, some patients may survive and recover, but others may experience long-term health problems, such as increased risk of cancer. Now, Hiroshima, Nagasaki, the people that were irradiated, they went through all of these different sicknesses. And they want to give them medical support like antibiotics. They just killed all their biotics. They want to give them more antibiotics. The bone marrow transplant, stem cell, yes. You got to replace, but what they really need to do is replace the bacteria. Now, what we need to do is to get a database of what bacteria is in the gut before and after a CAT scan. All right, and there's something called spectroscopy. I'm trying to get through a company that does this. And what we need to do is analyze the sample of the gut bacteria before and after a CAT scan. Does it affect it? And we should be able to tell quite easily. We won't know what exact species is being affected without doing some real deep research, but we should see that certain colors will be missing that were there before, or more colors show up. Something will happen to the spectroscopy, which is nothing more than seeing all these different signatures of light. It's bouncing off of something that's different. It bounces off this, it's different than bouncing off this. And the same thing with these bacteria. So if you see a lot of yellow and nobody else has yellow until they get irradiated, well, what happened? How come we got so much yellow? What's the yellow made out of? Can we isolate it? Can we ferment it? Can we look into it? What does it do to tissue? What does it do to blood? These are the kind of things we should be looking at. And this is radiation, is high dose of ionizing radiation, exactly what a CAT scan does. And this is exactly what, not everybody gets it, but it's not uncommon, I don't think. Okay, this is kind of shocking, really, I gotta be honest with you. This is microsieverts, 0.09 microsieverts per hour is the background radiation here. And that's the average and so forth. Now, so I'm a very low radiation area. Now, let's understand radiation doses, because that's the dose of radiation that I have to absorb. It is approximately 0.09 microsieverts. Now, we're talking about millisieverts. I'm talking about microsieverts. Now, there's other ones. You can have millisieverts. See this? For the general public, the standard annual dose limit is 1 millisievert, which is 1,000 of these. Only I'm way down at 0.04, 0.09 millisieverts. So I'm way down. So I'm really good to go, even if I was at 13, because every now and then it'll go up to 13. So I asked, is it okay for dose rates as 13 microsieverts per hour? And they say the annual limit is 1 millisievert. Well, let's take a look at a CAT scan. Okay, don't forget, we have, like here, I'm up to 0.14. It comes, it goes, you just never know. Now, a standard X-ray delivers about 0.1 millisieverts. This is microsieverts, so they're talking about 0.1. This is like 1,000 times smaller, because it's just every micro to milli is 1,000. Now, it makes the typical abdominal CAT scan the equivalent of about 100 chest X-rays. A CAT scan is about 100 of these 0.1 millisieverts. So it's 1 millisievert in one setting quickly. And when they use certain dyes, these contrast dyes, it literally doubles the amount of radiation exposure, because what's happening is when these X-rays are penetrating you, they're ionizing, which means they liberate other electrons, damaging the damage is done by breaking the cellular walls of the bacteria, killing the bacteria. The bacteria don't have a good defense against this really intrusive X-rays, and it kills them. So if you can't produce the enzymes because you don't have the bacteria, then you're going to be ill. You have to dispose of waste. You have to be able to attack the food that's coming into you. Otherwise, you get bloating, you get nausea, you get diarrhea, you get all the abdominal distress, really nasty stuff, gas, smells, terrible. And that's because you can't digest the food, because your gastrointestinal system has been damaged, which means your bacteria has been damaged. You don't have the enzymes. The enzymes aren't there, you're not going to get the chemistry. Enzymes work so fast, there is just no possibility of getting that job done without having that bacteria to create that enzyme. It's just not going to happen. But they can set up shop almost overnight, but you have to be flushed with those bacteria inside of you, and they pop right up and start doing their job very, very quickly. Okay, it appears to me they know there's risks and issues, but they say the benefit to risk ratio. Radiation dose is higher than a standard X-ray. The advanced imaging helps doctors diagnose serious conditions like tumors, infections, blockages that would not be visible on a plain X-ray. Potential benefits generally outweigh the small risk. Well, is it a small risk? Repeated scans. Over your lifetime, the risk from radiation can increase with repeated exposure over a lifetime. That's because you're killing off your bacteria over and over and over. And especially for children who are more radiosensitive. They're not more radiosensitive, they just don't have much bacteria. And the bacteria they have are not hugely populated yet. Their guts are just starting to develop an immune system. And you know, everything is discussed with your doctor, and you know, everything's fine, don't worry about it. Well, all I want to do, and it's very simple, it's no big deal. I want to see what bacteria and what populations of the microbiome were there prior to the radiation and after the radiation. That's all. And the radiation is the CAT scan. And they said it's 100 times more than a chest X-ray. This is as much as you're supposed to have in a whole year. 100 of these. That's one microsievert. And that's what you're supposed to have in a whole year, I believe. Yeah, I think I showed you this before. For the general public, standard annual dose, all year, a whole dose for a whole year, spread out over the whole year, is one of those microsieverts. It's for the whole year. And you get that in one setting while they're irradiating you. And for occupational workers, they're giving them higher limits. They let them get more exposed. The occupational, they have to be around these things, and they're so, you know, high energy that they're going to get exposed being around them. So you're getting the whole enchilada in one setting. You can lie down. I don't think that's good. And your bacteria, I would say, are going to get slapped around. CAT scans use significantly more X-rays, and it kills the bacteria. And the only thing that creates enzymes and ribosomes is bacteria. That's it. And enzymes are the only thing that does the chemistry in your body. That's it. All right, they know they're killing the bacteria. Food irradiation. Irradiation does not make food radioactive. Food irradiation uses ionizing radiation to reduce bacteria. It kills mold and pests and bacteria. The killing of certain bacteria by X-rays. This is 1930. X-rays kill bacteria. Bacteria create the enzymes to make your body work. All right, this is what I want you to focus on. Ribosomes. The bacteria create these ribosomes. In the bacteria, the ribosomes are floating around inside their body freely, but in our bodies, the ribosomes go inside of cells to protect them. So the ribosomes can be free in the cytoplasm, attached to the endoplasmic reticulum, or found inside mitochondria and chloroplasts. But they can be anywhere. So they can be in the cytoplasm, but mostly they're attached inside the cells. And every cell can have millions of these ribosomes. And these are the size of the cells. That's a cell in a body, a eukaryote animal cell. Okay? Now, you see this thing says ribosomes? Well, it's just pointing in it as they're all over the place. And there's millions of them, literally millions. And then you have this endoplasmic reticulum right here. This is where they collect. And I think it's like the gateways to get in to the central core. Those are the ribosomes are there to protect you. Ribosomes are killing factories. They break down food. They create all the enzymes. The ribosomes create the enzymes. I'll show you this right now how it works. Very, very elegant. Okay, remember I told you that you got ribosomes, which are these little blue balls. They're just floating around here in this, what they call the cytoplasm, just the fluids that are inside the bacteria. They just float around and do whatever they want. Well, they're actually being squirted out either on demand or just continuously. I think continuously. Now, this is the DNA that says I'm a certain type of E. coli and I produce a certain type of ribosome. And that ribosome, inside that ball, there is a set of chemistry that is so elegant, it's beyond belief. This little sucker can build that every time, and it has to be exactly the same chemistry. And when I show you the chemistry, it's just off the charts. And there's literally probably 100,000 or so different species. I think they claim now, I don't want to say, but I think it's about 75,000, something like that. And they're so nuanced that they create different breakdowns of chemicals in using half-lives. It just goes done. It does 1,000,000 years. One of these little enzymes, if it's present when you need to break something down, it will do 1,000,000 years worth of half-life breakdowns, because they're stable for a certain period of time, stable, and by eventually, sooner or later, 1,000,000 years later, it turns into aluminum, whatever it is. You need this, it goes done. And then it goes on to another one, another one, another one, another one, and another one. It's called click chemistry. And what does a ribosome look like when it unfurls? Well, here's what it looks like. It's got to come out of that sheath first. And that sheath is exactly identical to this situation here. It's got like a little rubber bag around it. And it just floats around until it embeds in a cell, or it can be free floating in the, it's just, they're everywhere. But primarily they're inside the cells. They say there's up to 1,000,000 in an actual cell. You see this? These are the ribosomes we're talking about. A mammalian cell, we're mammals, and we have cells, can contain up to 10 million ribosomes. Those are those little tiny balls, 10 million in the cell, and then you have all the other organelles in that cell still got to work. They're all over the place. It says a single bacterium, which is a single cell, may have 20,000 ribosomes, which that is what it uses to do its chemistry. But what it ends up producing is the enzyme that comes out and goes in and attaches in here. So the bacteria are the things that create the good stuff. All right, and they create these ribosomes, and you can have the eukaryote cells, which are in mammals and plants, typically have millions of ribosomes, up to 10 million or more in actively growing human cells. 10 million or more in one cell. Let me show you what a cell looks like. Don't forget, these things are like tiny, tiny, tiny. These are single cell bacteria. And they're all over inside your body. There's more bacteria in you than there is you in you and me. And when you poop, half of what you poop isn't even poop, it's bacteria. And it's used in your body to digest all your foods. They have to grow in copious quantities in good growing conditions, which means you have to have the right bacteria in there to create these ribosomes, to do the enzymes. And here's what happens. The ribosomes goes blip, blip, blip. And after it does the blipping, here's what happens. All right, so the little balls go squirting into the cytoplasm, which is just the fluids. And they float around, they go wherever they want. But apparently they either know where to go, or they're requested to where to go, or they just accidentally drift in. But they get inside the cell. This is one single cell. Ribosomes are in there everywhere. There's millions of them, up to 10 million, they said. Now, this is that endoplasmic reticulum right here. And they just, it's completely coated with this stuff, which it appears to me is the entrance to get into the nucleus, which is where the real chemistry is. That's where the blueprints are. All right, and this won't let you in here unless you pass through all the gates. Basically, that's how it works. So we've got a bazillion of these ribosomes in every single cell, and just floating around, just having a nice day. And if they're needed, bang, it's what's called a trigger. Well, what happens when the trigger goes off? All right, follow me down the lane. We gave birth to a ribosome, which there are bazillions of them in your body, really trillions and trillions and trillions. Inside of here, there could be millions in a single cell. And when they're activated, that ball, which I showed you, is this ball. It's like this. And it unsheaths, the wrapping comes off, and it does that. And why does it do that, Roger? Well, because every one of these balls is magnetic. And this is actually the magnetic signature. All right? This pops open. All that stuff goes, and it's locked in just like that. It's not, that's it, it's done. Once it's in its configured state, it matches up its dipole moments, which is all of those little signatures. You see these little dots? They're showing blue and red. Well, they're energetic little spots. And it's called click chemistry. It comes up to another molecule and says, hey, you're the guy that matches my signature. I'm supposed to kill you. Click, done. And then it goes click, 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 click. Instantaneously, I mean instantaneously. They say the only limitation to how fast an enzyme works is how quickly it can hit the substrate that it has to hit. So the quicker you rub it in there, the quicker that enzyme reacts with the substrate. All it's got to do is hit the substrate and it's done. It's instantaneously. And you see how elegant that is? If it's not that exact same way, it's denatured. It doesn't work. And I think it's probably, even if you just broke one of these right off here, that's it. It's done. It's got to be exactly that chemistry. And that opens up. Now, what does that mean? That means it can break down things. It can kill things. It can digest things. It can lock onto things and transport things using metals in the blood. There's a plethora of things you can do with that, just about anything. You can move things, you can break things, and you can build things. And they ask, are enzymes proteins? Well, traditionally, you would see them as two different things. An enzyme breaks things in my world. An enzyme is a product of a bacteria, and virtually all the enzymes come from bacteria. Those bacteria live in certain organs in your body that do secretions, and then they squirt out these enzymes. Now, the enzymes break things. Because you eat carrots and peas and corn and whatever you eat, meat, fats, sugars, carbohydrates, you got to break those things down into their molecular components. Really, basically, that's it, into their bits and pieces, which are these bits and pieces. And then you use those bits and pieces to create proteins. All right, so it's basically the same thing, but you're breaking this so that you can put it back together as a protein, which protein is a building block. It's your muscles, your tendons, your eyeballs. It's a protein is something used in your body, even like the conduction of nerves. Depends on the myelin sheath around it, and there's a lot of problems with nerve issues people have, and it could be related to these myelin sheaths. The diet we have now is not good. It's just not good, and it's killing the bacteria that's in our bodies, and people have been exposed to antibiotics, all kinds of things. Whatever kills your bacteria is going to harm your body, and broad spectrum bacteria destruction and 1 millisievert in a sitting, that's just, I don't think that's a good idea. But we don't know until we do a testing of before and after. What happened to bacteria. What happened to the bacteria in the body? That's the key. Now, I'm trying to get a hold of some people with spectroscopy expertise. I got some feelers out, but nobody ever contacts me back because I'm not a big anything. So if you know anybody that knows anything about spectroscopy, which means to take pictures of poop, and the CMOS can detect every single frequency in an exact frequency, and an exact frequency will mean that it's either a protein or what bacteria species it is, really right down to the species. And then AI should be able to look at that picture and say how much yellows are in here, and how much blues, and how much purples, and how much, all of these different colors are going to be different bacteria. If we find out we got way too much yellow after the scan or before the scan or whatever it is, we need a database. That's all. It's not a big thing. And spectroscopy should be able to do this, and you should be able to do it with a cell phone. Take the picture of the poop and just send it off, and you come back almost instantaneously with what the makeup of the poop is. And if you see yesterday you had the yellow was up here, everything was fine, and today you're bloated and everything's nasty and the yellow's way up here, well, whatever was keeping that yellow in check is gone, because we need a database. All of those things will filter out with the database. But if we kill the bacteria, we have destroyed the ability of the gut to do its job. Simple as that. And that bacteria lives primarily in this interstitium layer, which is right down here. And if you can get invaded in here, this is not a big deal. Right here, you get cut scratches every day. I get them all over the time. And you then go right down to here. That's okay. As long as these guys, and even you can go even deeper, where you can start getting pus and really red and nasty, because it's really attacking your good vital flesh. But these guys are supposed to come, and they will, as long as you have them. If you don't have the bacteria to create the enzymes, it's it. Stage 4 cancer. Cancer cells, they grow, and they're starting to eat and live in this area. And you don't want them there. Your body should have attacked them and sent them away. But it didn't, and eventually they will get through this layer, and your body will throw everything it can at that, and you'll have all kinds of, reactions in that area, possibly. So then they hit it with radiation and do all kinds of things. But I'm talking about a CAT scan right now, giving you enough radiation that I think is killing some of this bacteria. It's got to be. In my opinion, it's got to be. It's what it does. X-rays kill bacteria, and you're getting more than you should for a whole year in one sitting. I think it's got to be looked at, and at least do this testing. It's a very simple test. It can be done right now with a fecal swab, fecal matter before and after these medical interventions, and see what they do to the bacteria in your gut. We may just have to throw some more of that yellow back in there, or find something that attacks the yellow and brings it down, because it's going to be one way or the other. And these are what you have to have happen, and this is the chemistry, and it is elegant. And there is, like I said, millions of these little buggers in one tiny cell. So that's how much of a defense it can put up against invaders. And I really study the cellular nature, the tight cellular junctions, the adherons, and all everything. I studied it. You have to study every little bit in detail to do what I was doing with my fossils. This is the problem. Kill the bacteria, you kill the thing that's going to do this. Let me show you what they do. Okay, so what is a ribosome? I showed you it's these balls. Well, what's inside that ball? It's a program. This is nothing more than a program. The arrangement of these little polypeptides says, I'm going to do this, and it's, somehow it does it. I cannot explain it. It's extremely complicated, and there is 20 amino acids in each one of these little program codes. The elegance of the program is phenomenal. Now, you may have a short chain polypeptide, and it turns itself into a ball, or you see this? How they repel each other? All right, and all of that stuff plays into this thing. When it's inside that ball, it's in a ball like this. And it's completely surrounded. And these particles are so tiny that you can have 1,000,000 of these balls inside of another single cell in your body. It just staggers the mind. So here's the cell, the bacteria. We just created a ribosome. Here's what the ribosome is. All of these are amino acids. You see the different colors? They call it a polypeptide chain, and it squirts into a ribosome. And that ribosome programs messenger RNA. Now, I know you hear of messenger RNA, DNA. Messenger RNA is half of the DNA. It can splice into DNA with a molecule called uracil. And what happens is the bacteria creates a ribosome. The ribosome is it's a program. It's an extremely elegant program. And that comes out of the bacteria. And there's your polypeptide chain, and it transcribes using transferase. And it transfers that code into messenger RNA, which is a single strand. You see, this is the double helix. It's a double wind of it. Now, so what happens here? This messenger RNA, I don't know how it knows what to do and where to go, but it snips in here using uracil, the U. And it comes up with its set of amino acids to say, I'm replacing this program, or I'm building a new program, whatever it's doing. And that's polymerase. Anytime you see an ASE, it means there's an enzyme involved. And the enzyme works just like that. It cuts and it embeds it instantly. I would say instantly, pretty quick. And that is a process coming from the bacteria into the ribosome, basically, using this program codes, programming an mRNA strand, and there it is. And the uracil is the snipper that lets it come in, and now you're immune to whatever it is, or something's different in your body based on this enzyme. But you have to have the bacteria to create the enzyme. The enzyme comes out of the ribosome, which is the program. All right, so I showed you here, this is, the U is the uracil, and it cuts in to the DNA and allows the transmission of new codes. Basically, that's what it is. It creates new codes. It says genetic information transfer. In RNA, which is the one that went in using the uracil acts as a key base. It pairs with adenine from the DNA. You don't need to know any of this stuff. What it tells you is this uracil is found only in the RNA, and it opens up the DNA. Just think of it that way. You don't have to get too crazy about that. Now, something we also have to take into account, which would solve that problem of how viruses just take over and they change and they take over cells. The bacteria themselves have their own ribosomes. They don't just squirt them out. They do squirt out ribosomes, absolutely. But they have their own ribosomes as well for housekeeping inside their own cell. Now, I also believe that their own ribosomes program this cell to create a certain type of ribosome. So their own ribosomes have the program in it to make the ribosomes that they're going to shoot out. Something in their own ribosome changes. to say, I am going to shoot out something that we didn't shoot out before. And then that starts to program against you. Because they always talk about a virus just takes over a cell. And nobody could ever explain this. I think the virus itself can be propagated inside the cell of a ribosome within the cell, saying, no more making that ribosome with all that color. Put a pink over here and a black over here and a green over here and squirt that out. And that's what happens. Now, whatever the end result is, who knows? If it comes up and it invades your body, well, that's not good. If it comes up and it does something good for you, well, then that's good. But these are the programmers of your body. The bacteria makes the enzymes. That is the case closed. All right, so you know the bacteria create their own ribosomes, but they also create the ribosomes that protect you and flow through the system, which is our microbial biome, basically. And that's where those ribosomes exist. And they break down food and they do all those other things because they have the enzymes in them. When they're needed, they're there, and they're in copious quantities. So we know that happens. All right, this is really not that hard to understand. Bacteria are the biological factories. The bacteria is just like a factory squirting out these enzymes in the form of ribosomes, which are those balls. When they get activated or needed, whatever the trigger is, they pop open and do all that chemistry. Now, they're talking about culturing specific bacterial strains. Yes. Scientists can produce enzymes. Yes. We just need to have a database of what enzymes are good and which ones are bad, and what bacteria create those good and bad ones, and what happens to you when you get a certain biological interventions or medical interventions, different inoculations and antibiotics and vaccinations and radiation and all that stuff. All I'm saying is just take a test. I'm not saying do anything medically. We just need to get a database. All right, there's a whole bunch to read about this, but broad spectrum, that means looking at all the different colors that bounce back. Ultimately, broad spectrum spectroscopic methods, especially when combined with advanced analytics, which is the AI, offer a promising path for rapid, label-free, accurate identification of diverse bacteria in biological samples. All right. Now, what you need to do is to have a technique to prep the sample, because it's tricky. That's one of the problems, is getting the surface consistency, interference from the matrix, challenges. But if you can see what's in that smear, and there's got to be some way of taking like a tiny little block that has tiny little divots in there and making a smear on there, and then they can analyze all those different little spots with the cell phone, send it off and have it come back and hopefully get some kind of results. But there's going to take some figuring out what to do here. But if you can use this to identify the bacteria, and you can, they say that's not a problem. You can find the bacteria. That's no problem to do that, the identification. But it's see this how it works for bacterial identification. The signature is determined by its unique chemical composition. Every single living thing has a unique chemical composition which gives off a different frequency of energy. Each type of spectroscopy captures.
This is a
all right, just to tell you right here, it says when combined with machine learning and advanced data analysis, these techniques can differentiate between various bacterial species and even the strains of those species. The exact bacteria. Now, how this works for identification is the bacterium's spectral signature is determined by its unique chemical composition. Every living thing has its own exact signature. Each type of spectroscopy captures the unique vibrations of the chemical makeup of the bacterial cell, which can be used to classify it for classification. This can be done. There's some work to do to figure out all the little nuances to it, but in the meantime, you have to understand these enzymes are built from the bacteria. All right, it's not. We need to know what bacteria is missing, because these people have these long COVIDs. They have these, all kinds of issues, the chronic issues that never go away. You're never going to have that issue go away until you put the right bacteria in there to have the right enzymes. It's just that simple as that. Simple as that. And the people that are very healthy, I would assume they have they poop regular. They don't have a lot of gut distress. There's a whole set of issues that goes along with bad digestion. The gas, the smells, the, irritable bowel, bloated, nausea, diarrhea, constipation. All that stuff is an indication you're not digesting something correctly. And it may not be a whole lot of things. And then there might be something that is growing inside of you that shouldn't be there because you don't have the right guys to fight back against them. Everything's supposed to be a balance. And when you say everything's working right, it is. So what we need to do is get a database of before and after these medical interventions or CAT scans, all that stuff, just to find out what is basically a healthy gut. Now, something you have to take into account is where you live, what the water you drink, the air you breathe, the radiation in the background. See, my radiation is very, very low here. All of these things play a part in how you react to your environment. And everybody in that area may be subject to bladder cancer. For what reason? Who knows? The water has some extra molecule in there because that's what's going to happen. But if you knew that nobody else over in this area got bladder cancer because they didn't have any of that pink stuff way up here, well, what's the pink stuff? And how do you stop the pink stuff from going way up there? These guys don't have it. Why? Is there something in that water? What's in that water? What's the bacteria? Because the bacteria make the enzymes do the chemistry. They do the bad chemistry, they do the good chemistry. Simple as that. And the way the body is designed, you're supposed to have enough good chemistry in you to fight back against the bad chemistry. And they breaking into your body, you only have a little tiny spots that they come in. And your whole body is filled with fluids. There is so much lymph fluid in you. There's more lymph fluid in you than there is blood in you, I believe. And it all carries these enzymes and bacteria, which are your emergency guys, and they break down your foods. They do all that stuff, all the things I've been talking about. So you should be able to stay healthy by keeping that system healthy. And then when you're not healthy and you're having all those symptoms, you should be able to take a picture of your poop, send it in, and it'll come back and say, you're too high on this and too low on that. And it might be able to make a recommendation. It may even just send you the damn pills. Who knows what you can do with this? But before we can do anything, we have to understand why are some of these kids getting autism and other ones aren't. And I can tell you right now, they're using live viruses. If that kid has no possibility of fighting back whatsoever, and he gets a live virus, there's a good chance he's going to get some kind of a reaction. I mean, it's just the way it is. Live viruses, if there's nothing at all to fight back, and they assume you're going to have some reactivity to it. If you have no reactivity to it, then you got a problem. And I believe that's what's happening. And you could easily find out by what the kids have in their bacterial count before and after. And if you keep finding the ones that having emotional issues and this kind of problem, autism, it'll work its way out, and you'll be able to figure what's causing it. And you may be able to just supplement it. Right now, we've been helpful at autism with just probiotics and a couple other simple chemistry right up in that membrane area, which is what the problem is, in the phospholipids. And Marguerite, who went to Harvard at the same time I went to Johns Hopkins, we worked together with her son, who was off the charts autism and is doing fabulously now. And we realized that, well, we worked on the fact that there was phospholipid membranes. The membranes protect you. And she worked on both sides of this, the silicon and the sulfur. She's got some kind of little mix she's using. And then all these probiotics. And the probiotics create the enzymes. And with these extra little chemicals up right in that phospholipid area, that protects you from invasion and makes the phospholipids turn slippery and everything else, I believe. Because you see this here? This is synovial joints in humans, and I'm sure that's phospholipids. They're like way off the chart slippery. And you have to have those phospholipids, and you need these sulfides and the silicon right up in that area, because they work with those in half-lives. If you look up the half-lives of phosphorus, you're going to see they go up to sulfur, and they go down to silica. And that's anyway, it's a little tricky, but she worked it out, and he's doing fine now. John is in very good shape now. He's at school. He's what she says, he's at the top of his class. I don't know whether she's just bragging or not, but but he's talking. He was never, not a word he ever said. They were going to put him in a locked ward, they call it over there in Belfast, Ireland, and they cut the kid's guts out. Well, first thing I said to her, how's he poop? She says, oh, every one of them has the same issue. They have digestive problems. I said, no. And she thought it was because of the autism. I said, no, they're not digesting correctly. That's what's giving autism. And she said and I didn't do anything with her. She did it all herself. I discussed things with her, that's all. I'm not a doctor. I don't give medical advice. I just followed her, what she was doing, and she saved her son from going into that locked ward for the rest of his life. For the rest of his life. And they told her there was nothing could be done with him. He was so far off the charts, they wouldn't even let him participate in a study. 9.5 years old, never said a word in his life. Biting and hitting, and, they were afraid for his own safety and the safety of other people. He just couldn't control himself, stimming and humming and all of the nonverbal and totally not communicating with his body. And that's really what it ended up being, because he could remember stuff, going to the zoo and stuff. She took him to the zoo when he was in this condition where he was like he wasn't even there. You wouldn't know he was there. You'd look and say, the kid's gone, he's not even there. Well, he remembered where the giraffe was and where the cage was, and they moved the cage because they went to the zoo after he was better. And he said, wow, look, they moved the giraffe. And she said, you remember that? And he said, yes. Wow, my hands, my hair is standing up on my arms. I'm telling you, the rest of his life in a locked ward, and he's doing very, very well now. That's my understanding. And she's got this autism group going on Facebook. Ascension Autism, I believe, is the name. I'll have to get that for her. Hopefully she'll comment on this, and I will pin that. All right, there it is right there. Autism and gut health issues. Marguerite O'Reilly, she does the group. And you can go up there and discuss back and forth. What worked for you, what didn't work, what helped, all that kind of stuff. It's a forum to speak about her experiences and, your own. This is a tough place to be in that, the family is very, very seriously affected. The kid is affected terribly. And if it's not necessary to have this much of a distress, well, it seems pretty obvious to me, you would want to do some investigation. This is not a big, long thing you have to go study for years. We have, but you don't. We're just going to show you what the research shows, and then you just make your own decisions from there. But I find that when you do have some knowledge about some things, then that leads to your own examination of what is actually true. That's the most important part. What is actually true?